Okay, so good morning, everybody, or good afternoon, whenever you've chosen to watch this recording. And today we're going to start discussing the membrane and membrane structure and membrane function. So you'll realize over and over again in class, I've emphasized the importance of the membrane itself, both the cell membrane around the outside of the cell, but also the membranes which constitute the organelles as well. And the singular importance of the membrane being that it allows control of conditions on one side and the other, so that conditions on one side can be different to those which are present on the other side. Today, we're going to learn a lot about how that control is exerted. So to remind us, we did discuss this when we discussed biomolecules, but just to remind ourselves, the membranes are comprised, the basic units of the membrane are comprised of these molecules called the phospholipids. And phospholipids consist of these long hydrocarbon chains, the fatty acid chains there, which are lipid. And then they have a head region here, which is a couple of things, but the really significant thing is this phosphate group in the middle here. And the combination of those things, phosphate group and the lipid, lead us to call them phospholipids. These long lipid chains here are basically strongly hydrophobic. They're oil, so they don't dissolve in water. They repel water. These heads here are highly soluble in water, so they are hydrophilic. This kind of schizophrenic nature of this molecule is, in formal terms, we say that it is an amphoteric molecule. And that means it has a dual nature. It has a hydrophilic part and a hydrophobic part. And think back when we talked about the biomolecules, I showed you that if we force a whole lot of these molecules to be immersed in water, then there's only one way that they can behave so that they satisfy both the hydrophilic and the hydrophobic requirements of the molecule. And that is to dissolve the hydrophilic heads in water, in the aqueous medium, there and there, and to sandwich these lipid fatty acid tails in between there, so that this area here is a strongly hydrophobic and it excludes water. But on the outside here, these are hydrophilic and they are happily dissolved in water. This is essentially a liquid barrier. The molecules are associated with one another by very weak bonds, mostly hydrophobic bonds, which are extremely weak. And as a result, these molecules are actually quite mobile. And this really is a fluid. It's a liquid, but nonetheless, it forms a very powerful barrier. And it forms a barrier to the passage of substances across, especially because most of the substances that we're going to, things like ions, this sort of thing, anything that is polar finds it very difficult to move across this hydrophobic area here. There are some molecules which can get across. Gases can get across quite easily. Some ions can move across slowly. Even water molecules can move across, but they can move across extremely slowly. There is not an efficient transport of these molecules across. These lipid tails here are not all of one kind. There can be several different kinds of fatty acids involved in construction of these phospholipids, and different lipids have different qualities, different properties. And if we vary the amount of a particular lipid in a membrane, we can actually change the nature of the membrane, as we'll see in a second. So that is the basic structure. It's a double leaflet one there and one there, of these amphoteric molecules, which are partly hydrophilic, partly hydrophobic. The key to the functioning of this membrane is that the whole membrane is actually, it is semi-permeable. It allows some molecules to cross it easily. It does not allow other molecules to cross it. And therein lies the control over the functioning of the whole membrane. But we need to remember that it does not just consist of these lipids. There are many other components to the living membrane. This phospholipid skeleton is actually modified by the addition of many other substances. And those in particular are the proteins which are associated with it. There are many proteins associated with the membrane because proteins perform so many different functions, and many of them are membrane associated functions. There are two basic classes of proteins which are associated. The first are called the peripheral proteins, and here they are here. This is the cytoplasm on this side. These are peripheral proteins. They are proteins which are associated with one side only of the membrane. Most of them associated, in fact, with the cytoplasmic side. There are a few which are associated with the outside, but the cytoplasmic peripheral proteins are much, much, much more common. So these are just associated there, and they tend to be associated with these hydrophilic heads. There are a whole other group of proteins, however, which are embedded in the membrane. They may be partially embedded, like this one here, for example, there. It's actually protruding on this side, and it's embedded there, but it's not protruding that side. This one here, on the other hand, these ones here they go right through the membrane. They emerge here, they emerge here, and they span the whole width of the membrane. Both these and these are called integral proteins. And there's something really important about them. In order to be embedded in the membrane in this way, the outside of these proteins, what is facing to the outside, has to be compatible with their location. In other words, this whole area of the protein here on the outside of the protein must be hydrophobic. It can't be hydrophilic because it's in a highly hydrophobic environment. So the center part of the protein is hydrophobic, and it anchors it in the membrane in a way. These parts that protrude into the cytoplasm or into the medium or whatever around the cell, these are hydrophilic because they're in an aqueous environment. So remembering the structure of proteins that we talked about before, you'll realize that these have to be folded very, very carefully. They have to, we'll see it again in a minute. They have to be folded very, very carefully so that they preserve the compatible structure to sit in the membrane. So we refer to this whole system as being a fluid mosaic. It's a mosaic because there's many different molecules that have been put together to form the whole. It is a fluid because it literally is fluid. This whole phospholipid skeleton of it is actually a liquid, and these proteins can actively move. The lipid molecules themselves can move, but the proteins can move quite extensively, and they can be moved. Remember what we talked about before about the motor proteins operating together with the cytoskeleton. They can actually grab these proteins and move them around inside the fluid phospholipid. If a particular type of protein is needed in one part of the cell or another, it can be actively moved. They can be rafts. We visualize them as being like rafts that can be moved through the fluid of the phospholipid. So this is a highly mobile, very, very dynamic system. And it's the nature of the proteins which are embedded in it, which give it its living quality. The other thing I just want to remind you about is that many of these proteins that we see on the membrane are also associated with the cytoskeleton. I've just mentioned how the proteins can be moved. Well, these here underneath, which are microfilaments of the cytoskeleton, these are actually actin. And they can be associated also with the motor proteins. And that is how we can move these proteins around. But these proteins are also anchoring proteins, which anchor the cell membrane to the cytoskeleton on the inside of the cell, and then to all of the extracellular materials, the collagen, all the glycolipids, all sorts of things that occur on the outside to stabilize the cell and the cell membrane. Okay, so I've several times now emphasized to you that this membrane is actually fluid, and it has to be fluid. It has to be fluid in order for it to work properly. If we affect the fluidity of this membrane, we're going to alter both permeability and then the actual ability of the proteins to move within the phospholipid background. So these molecules are all mobile, but it is essential that a certain level of fluidity be maintained. And hold on, I'm missing just 1 second, please. I'm sorry, I missed a diagram. So it's essential that the fluidity of this membrane be maintained. And the fluidity is determined by the types of molecules that are present. Think back to when we talked about hydrocarbons. And you'll remember that I told you, if a hydrocarbon chain is saturated, if it's got the maximum number of hydrogens associated with that can be, then that molecule tends to be straight. Here is phospholipid. All of this phospholipid has straight, saturated fatty acids associated with it. These are much more viscous. They tend to be much more viscous because the weak forces holding them together are distributed all through here. So although the forces are weak, there are many, many, many of them. And these tend to be much more viscous. You'll remember that, in fact, saturated fats tend to solidify at lower temperature. It's exactly the same in this system here. Lower temperature, as the temperature drops, they tend to get more and more viscous until they become non-functional. The way in which the cell regulates the fluidity is twofold. The first is by regulating the number of saturated fatty acids. If we include unsaturated fatty acids, the unsaturated tails have a bend in them, and they prevent the molecules from packing together and establishing the maximum number of weak bonds. Instead, they are kept apart from one another, and this is much, much more fluid. And the cell can rapidly alter the proportions of types of fatty acids in membranes in response to environmental temperature, for example. We don't have to worry about it so much because we maintain a constant body temperature, and this is one of the reasons why that's advantageous to us. We don't have to, our cells don't have to worry so much about this. The other way in which fluidity of cells is maintained is by packing this molecule, which is also a lipid, but it's a steroid, and that is cholesterol. And cholesterol is an essential lipid in our bodies, and this is one of the reasons why it is used to actually modify the fluidity of our membranes. And these here are much more fluid than these here, which lack the cholesterol. Okay, so that's the basic structure of the phospholipid background. Now we need to spend a little bit of time talking about membrane proteins and of their different functions. I've explained to you that peripheral proteins are associated with this and bound to the surface of the membrane. Integral proteins have some part of them embedded in the hydrophobic area of the membrane, as well as being in the aqueous. And they may span the entire membrane. If they do span the membrane, they are called transmembrane proteins. And these are integral proteins that go from one side to the other. And as I explained to you, they have a hydrophobic region here so that they are compatible with the hydrophobic areas of the membrane. They have a hydrophilic area, which is here and here, so that they are compatible with the aqueous medium in which they are embedded. So immediately realize these proteins have something singular about them, and that is that they communicate. They communicate one side and the other. Now, that one side, that may be the outside of the cell and the inside of the cell, let's say. That's what this diagram is showing. But the same principle applies to proteins which are integral to all the other membranes in the cell. There are many, many other membranes. There are membranes around the organelles. We saw that mitochondria and chloroplasts have much folded inner membranes, which also have their own peripheral and integral proteins. And in all of them, the importance of the integral protein is that it communicates between one side and the other. And that allows for the possibility of many different of these proteins performing many different functions, as we'll see in a minute. So here are the principal functions of these transmembrane proteins. The first is that, and we'll think about this in some considerable detail, because this is one of the most significant of all of the functions of these membrane proteins, and that is in transport. That is in transporting compounds across this membrane, remembering that this membrane is impermeable to many, many things. It is particularly impermeable to things like ions, and also to some of the larger molecules. It's even relatively impermeable to water. Water only moves across slowly. And all of these things may need to be moved across very, very rapidly. Sugars, all these sorts of things are constantly in demand by the cell and need to be transported. And it's the responsibility of these proteins embedded in the membrane to do so. There are two, and we're going to see that there are two different ways in which these proteins can operate in order to do this. The second set of functions of these embedded proteins is that many of them are enzymatic. And we're going to learn, for example, when we talk about respiration, when we talk about photosynthesis, these kinds of processes are membrane-associated processes, and they depend upon these proteins being embedded into the membrane. And it is the presence of the membrane and the associated protein that allows the functions to occur. They wouldn't occur otherwise. The third function of these proteins, remember I said that they communicate one side and the other. So these proteins can in fact relay information from one side to the other, and they do. This is called, this system here, there are different systems, but this is the system that we'll learn about, which is a signal called signal transduction. This protein here, spanning the membrane actually has a receptor site. And that receptor site is very specific for one particular thing, one particular substance. It can be something as simple as an ion. There are some which are sensitive, for instance, to the number of protons and signal the pH, this kind of thing. But there are others which receive distinct signals, such as, for example, a nervous signal. This is one of the ways in which nerve impulses are relayed. There are proteins embedded in the membrane. They have a receptor site which binds one particular substance. And when that substance binds, they don't transport the substance across. They don't need to, because this is just a signal. It is to inform the cell that it needs to do something in response to the presence of the substance. So what the binding here induces the protein to signal to the cell that some particular response is required, often a metabolic response, for example. There are many examples of this. There are many proteins which are sensitive to particular chemical substances. There are proteins which are sensitive to sugars and signal that sugar is present in the environment. There are the nervous, which respond to neurotransmitters, all of these kinds of things. But there are also proteins which are sensitive to things, for example, like growth hormones and that sort of thing, to other hormones, which when the hormone is present, signal to the cell that the cell needs to begin behaving in a particular way. Okay, so these are the signal transduction proteins. They are really the eyes and the ears of the cell. They relay a huge amount of information from the outside to the inside of the cell. There's also, when we start to take cells and we start to associate them with one another to form tissues and organs, for example, we actually need to have the cells be able to recognize the presence of other cells that they need to be associated with. And this is done in particular by labeling some of the proteins. Very often proteins are modified by adding sugar groups to them. And these are the glycoproteins. These you'll recognize. These are little these are little carbohydrate chains rather. These are all glucose units here. And this is a glycoprotein. It's a protein which is modified by the addition of sugar groups, often as a label. And specific cells have specific glycoproteins that are present. Those glycoproteins can be recognized by other cells. And that will induce these cells to do something. For example, it may end up with the cells joining. Other joining proteins are used to actually tie the cells together. I told you before about things like tight junctions, this sort of thing. All of those things which tie cells together into a tissue and then the tissues together into an organ, all of those depend upon cell-to-cell recognition, upon intercellular joining, and lastly, to something that we've already heard about. And that is there are specific proteins embedded in the membrane which are responsible for attaching the membrane itself and hence the whole cell to external proteins and elements which provide cell shape and cell stability. And in addition, they attach to elements of the cytoskeleton on the inside of the cell. So there's 6 major functions of these integral proteins. All right, so I've said to you several times that the membrane is in fact selectively permeable. It will allow some things to go across quite easily. It will exclude other things. The basic nature of the membrane, the phospholipid membrane, is actually exclusionary. There's not a lot of things which will get across easily. as I've told you, gases, some very small molecules and things like that may make their way across, but usually they do so very slowly and far too slowly for it to be useful to the cell or to its metabolism. Instead, the cell controls what passes across one way and the other. And this is what allows the cell to keep conditions at optimal inside the cell relative to the outside. It is the activity of the membrane itself, and that activity is governed by the nature of the phospholipid background, but largely by the activity of the proteins which are embedded in the membrane. It's the proteins which are actively determining what goes across and in what direction. So the only thing which larger molecule which gets across relatively easy are things like hydrocarbons. And that is because they can cross that hydrocarbon barrier quite easily. That hydrophobic layer on the inside is really what provides the largest barrier to the movement of molecules across the membrane, because it excludes hydrophilic things, polar charge molecules. All of these things find it very difficult to move through a hydrophobic environment. But the cell needs all of those things. The cell needs hydrophilic molecules, many of them. It needs polar molecules, it needs water for a start. It needs charged molecules, it needs to control the passage of ions across that membrane. We're going to see there are many circumstances where the cell needs to accumulate molecules for use. It needs to control the concentrations on one side or the other. And all of these things are done by the proteins which are embedded in the membrane. And the reason for that is that these proteins can act as transporters. And they transport under very special conditions. In general, they transport anything which is hydrophilic. Hydrophobic substances, which are really needed by the cell required, may pass across quite easily. But any hydrophilic, polar, or charged substance finds it very, very difficult to do so. And instead, there are proteins embedded in the membrane which act as channels to allow these molecules to go through, but to go through under very controlled circumstances. These channel proteins are highly selective. So they don't just there's not just a tube embedded into the membrane. They are very selective about what they allow to pass through. And so they can exert tremendous control over what comes across. So those are the channel proteins. They are proteins which act as a channel, literally, for specific substances to move across. There is another whole other set of proteins and integral proteins, which are carrier proteins. And they are called carrier proteins because that is literally what they do. They will bind a substance and they will change shape and move it in a specific direction. We'll see how this works in a minute. There are some specific channel proteins, transport proteins, which we need to pay particular attention to. One of them is the glucose transporters. And this is an example of how specific these are. Glucose transporters will transport glucose and glucose only. They won't transport even things which look very, very much like glucose. Fructose, which is an isomer of glucose. It's the same chemical formula and everything. It's practically the same shape, but just a little bit different. It cannot go through the glucose transport. It needs its own transporter in order to be transported across the membrane. The one set of carrier proteins, of channel proteins, however, that we're going to pay particular attention to are called the aquaporins. The name tells you what they do. They transport water. They allow water to move freely across the membrane. And these are channel proteins, very specific for the water molecule itself. And they are the principal way in which water will move across. And it's one of the reasons why the membranes are semi-permeable. They are permeable to water, but they will exclude anything for which there is not a channel protein and which cannot freely pass across the membrane. Okay. This is so the whole nature of the membrane, of what it allows across, how it allows it across, depends upon the proteins that are embedded in it. So we need to spend a little bit of time thinking about how molecules actually move back and forth across a membrane in general. So here are some theoretical experimental situations. And we have here a membrane. This is a membrane actually is going to allow substances to move back and forth quite easily. And we just, on one side, we are going to put a whole lot of a substance, a dye or a salt or something like that. When we start off on one side of the membrane, there's lots of that. On the other side, there is very little. It is a physical and chemical law that the substance will attempt to distribute itself until it is evenly distributed throughout the entire space. This is because all of these molecules are smacking into one another the whole time. They have a certain level of kinetic energy and they are moving. And if you think about it, when they are smacking into one another, where is their net movement going to be? They're going to tend to be knocked out to the areas where there are no other molecules to knock them back in again. So their net movement is going to be in this direction. This is not to say that they are not moving in this other directions. They are, but their net movement is towards the area where there are fewest of their own molecules. And so this is the process that we call diffusion. Diffusion occurs through any medium where the molecules can move freely. It occurs in gas, it occurs in water, etc. And if we have lots of molecules aside, they will tend to distribute themselves in this direction. They diffuse, and net diffusion is in this direction. And we say that it is down the concentration gradient, from high concentration to low concentration on the other side. If we leave it long enough, then what is going to happen is we're going to have equal numbers of molecules on this side as we have on this side. That is now a situation where the molecules don't stop moving. They don't stop being diffused. They don't stop being knocked around by one another and driven in different directions. They are. But when they reach a situation where there are as many here as there are here, if one more moves this side, then one other one will move that side. Because if I move one across here, I've increased the concentration here and I've lowered the concentration there, and the system will even it out by diffusion. And so when the system reaches equilibrium, as many molecules as pass in this direction will pass in that direction there. The way to really think about this is, and this is actually a pressure. It's a diffusive pressure. This substance in high concentration is exerting a pressure, and it is moving molecules in this direction. It is moving molecules down the concentration gradient. So that's our first concept. Our second thing that we need to think about is, what if we have a situation where we've got two substances which are in the solution? Let's say we've got one on one side and one on the other. In actual fact, those substances will behave independently of one another. They will behave as if the others were not there. They will diffuse this one will diffuse that way, down its concentration gradient. This one will diffuse down its concentration gradient. And until they reach an equilibrium, it's called a dynamic equilibrium. The equilibrium is not static. If we knock one in one direction, then one will move in the other direction to maintain the equilibrium. So in this case, we've got two solutes, one on this side, one on this side. They behave entirely independently of one another. This is really important because what we are actually talking about when we talk about substances in solution, we're talking about two things. We're talking about the solute, the salt or whatever that we've dissolved, but we're also talking about water molecules. And we need to think that there is actually a concentration of water as well. And I'll explain more carefully what I mean. This membrane here is not selectively permeable. It's not making any choice about what can get one way or the other across this membrane. It's this membrane that we've been talking about here. It allows free movement. But many membranes are semi-permeable. That means certain substances can get across, but other substances cannot get across. And that's the situation that this experimental system here describes. Here's the setup. We've got a U-shaped tube here, and here we have a semi-permeable membrane, a selectively permeable membrane. And what it is selecting is that it's going to allow water to pass across it quite freely, but it's not going to allow salts and things to pass across it easily. So here we have our experimental setup. They've used here sugar, and they put a high concentration of sugar on this side of the membrane here, and a low concentration of sugar there. So how is this system going to behave? Well, if there was no membrane there, then what would happen would be that the sugar would distribute itself evenly throughout. But that's not our experimental system. Our experimental system has put a semi-permeable membrane here, which allows only water to pass. But here's the thing you need to think about. What is the concentration of water on this side and the concentration of water on that side? On this side, we've got masses of sugar molecules. It's the same volume. We start off with the same volume. And in that volume, we've got masses of sugar molecules. This side, we have only a few sugar molecules. What's the concentration of water molecules on this side? It's much lower than it is on that side. And water diffuses just the same as any other molecule. The diffusion of water, we refer to by a special term, which is osmosis. And what is going to happen now, purely because we put a semi-permeable membrane here, these sugar molecules cannot move across here. They cannot move across to equalize. They're trapped behind here. But the water molecules, this is a semi-permeable membrane. It's permeable to water. And the water will move down its concentration gradient. Water concentration here is low. Water concentration here is high. The water will move across here, and lo and behold, the volume will increase. We'll lose volume here as water moves across here. And this is what you need to think about, that this is very much like the cell. Because the cell membrane, for example, does not allow the free passage of substances like sugars and ions and things across the membrane. It just doesn't. They can't get across. But the cell membrane has many of these channels which called the aquaporins, which allow water to move freely. And as a result, water tends to move into cells very rapidly through these aquaporins. And what it is doing is not moving through to dilute the salts inside. It is moving through to equalize concentration of water molecules on one side and the other. That is the actual mechanism for osmosis. So these here describe this to you. Okay, here are the sugar molecules. They cannot move across. These are free water molecules, and they can move across this membrane because it is semi-permeable. And they will attempt, they're going to move down their concentration gradient and attempt to reach the same concentration. There's another little flip to this, because in fact, the water molecules tend to bind to the sugar, and it is the free molecules of water which we need to worry about. So there's an increased pressure in that direction due to osmosis. Okay, so this basically just explains to you the entire system, and that is this side here has a very low water concentration for two reasons. First of all, there's lots of sugar molecules there. And second of all, because the water molecules tend to cluster. Tend to cluster to the sugar. Water tends to move in this direction, and not so much in that direction. Okay, this has very important implications for living systems, because cells are immersed in fluid. The basis of all life on Earth is actually aquatic. All of our cells are immersed in aqueous media of one sort or another, of varying concentrations of solute, and also environmentally very different environmental circumstances can arise in which cells must live. So let's take a situation first where we take a cell, remembering that the interior of a cell is usually very rich in solutes and molecules. And so it actually has rather a low water concentration. We're going to take these cells and we're going to immerse them into a solution which is very much lower in solutes around them, which obviously has a high water concentration. And that situation we call a hypotonic, a hypotonic solution is, we are saying that the exterior here is hypotonic to the interior of the cell. These hypo, etc. always specify what is relative to what. Okay, you always need to specify it's hypotonic to. So the exterior here is hypotonic to the interior of the cell. What is going to happen? The concentration of water out here is much higher than it is inside the cell. Water is going to flood into the cell through the Aquaporin channels, and the cell is going to begin to expand. The volume inside is going to increase just the way the volume here increased. And this is a very dangerous situation indeed, because if you may do this in lab, I don't know if you will, but it's commonly done in lab, that you immerse red blood cells in distilled water or whatever, not in the saline that they are normally, that is present in our blood. They very rapidly swell and burst. It's a grave danger for cells which are in hypotonic solutions. And there are many cells which are in hypotonic solutions, as we'll see in a minute, and they have to do something about this. The simplest solution to this is present in the plants and in prokaryotes, and that is to have a cell membrane, sorry, not a cell membrane, a cell wall around the outside of the cell membrane, a strong, rigid cell wall against which the cell, when it swells, can push. And the pressure of water needing to come in is called the osmotic pressure. It is a real pressure. And the resulting pressure inside the cell, as the cell swells, is called the turgor pressure. And the turgor pressure presses the cell up against the cell wall until the turgor pressure equals the osmotic pressure. In other words, the pressure, the impetus for molecules to move in through osmotic pressure is counterbalanced by the turgor pressure pushing back. And at that stage, equilibrium is reached. The cell is placed under pressure. That is true, but that doesn't really affect the functioning of the cell. We reach a system of equilibrium here as many molecules pass one way as will pass the other. That's the typical dynamic equilibrium, when the two pressures are equal to one another. If a molecule passes this way, then one molecule will pass that way. Why? Well, if I let a molecule pass across here, I've increased the pressure in here, and it's no longer equal. So a molecule will pass out to equalize the pressure. This is a simple system of osmotic control used predominantly in the bacteria and in the plants. And in plants, there's another slight complication, and that is that they regulate it by means of this large organelle here, which is the central vacuole, which is an aqueous solution of salts. So with a very low water concentration, so that water continually moves into the plant cell and creates this pressure against the cell wall. Okay, so that is when we have the cell in a hypotonic solution. What about if we put the cell into hypertonic? Here, the outside has a very much higher solute concentration than inside the cell. In other words, it has a much lower water concentration outside here than inside. And water now floods out of the aquaporins. The aquaporin channels are simply a channel. All that they regulate is the type of molecule that goes across, which in the aquaporin case is water. They do nothing to regulate which way it's going at all. So the aquaporins now allow water to flood out of the cell, and the cell will begin to shrink. And if you do this with red blood cells, this is what they look like. They end up with, we call this hemolysis. They end up just shrinking up into these little tiny biscuits, and the cells shrivel. Now, a similar situation can arise in plants, and it arises under one particular condition, and that is when you forget to water your plant, or when there's a drought, or the plant dries out. What happens if a plant dries out? What happens to the water concentration? The water concentration around the cell falls because the plant is actually actively drying out. It's losing water molecules. And as a result, water begins to flood out of the cell. And when that happens, the whole plant cell begins to shrink like this. It shrinks away from the cell wall, and we call this situation plasmolysis. Plasmolysis, the cell begins to shrivel and move away from. You can do the same thing experimentally. If you take these plant cells, for example, and you put them in, take a freshwater plant, for example, and put it into salt water. And this is exactly what happens. The cells begin to shrink away from the cell wall and plasmolyzed. In isotonic solution, an isotonic solution is one where the concentration of salt and the concentration of water is equal on either side of the membrane. In that situation, it's not that no movement takes place across the membrane. It's that as much movement takes place across the membrane in one direction as takes place in the other direction. if I watch one molecule of water move across that in an isotonic situation, one molecule of water will move out, because if I allow one to move in, I've slightly increased the osmotic pressure inside. And because of that, one molecule will move out to equalize the concentration of water outside as inside. So in most situations, this is a very healthy situation. There's one set of organisms don't particularly like this kind of situation, and those are the plants. Plants actually function best when they have this high turgor pressure inside the cells. And at this, in an isotonic situation, plants are right on the edge of beginning to wilt. They actually require a high turgor pressure in order to maintain their structure against the force of gravity. We say that these cells are flaccid. They prefer this turgid situation, which is the normal situation. Okay, so first of all, here we've talked really as if everything is just relatively passive, as if the cell, there's not a lot that the cell can do. The plant and prokaryote cells, they have a strategy, and that is to have a cell wall, which affords them osmotic control. But most other cells actually need to actively control the movement of water one way or the other. And because they can't tolerate taking up too much water, they can't tolerate losing too much water. So they regulate what happens. And we refer to that as Osmo Regulation. And especially in aquatic environments, Osmo Regulation is a critical feature of cell metabolism. There are some situations which are really, really dangerous and difficult. And surprisingly enough, living in fresh water is one of them. Living in a desert environment where there's very little water is also extremely dangerous. But quite honestly, living in fresh water in many ways is almost as difficult as living in a desert environment. And the reason for this is that was something I've already told you. The cytoplasm of the cell is very rich in solutes. So in actual fact, the cytoplasm of the cell is always relatively poor in water molecules, usually compared to the outside, in almost every situation. And especially in fresh water, there is a huge pressure from outside, osmotic pressure exerted on organisms which live in fresh water. One organism you've heard about is Paramecium. Here it is here. Many single cell protists which live in fresh water adopt this particular strategy. Water continually floods into them, and they have to deal with it the whole time. And they do so by pumping it out. And they pump it out with a special organelle called the contractile vacuole. Here it is here. These here are actually channels associated with the contractile vacuole. They feed water into the central vacuole and into the contractile vacuole. And the contractile vacuole empties to the outside. And it does so with regularity, really every 10 seconds, 15 seconds, whatever. You'll see a contractile vacuole filling and then emptying to the outside as they pump the water out. These Paramecium actually have two contractile vacuoles. One is filled like this, and as this one empties, the other one will fill. And you can see them kind of going like this, emptying to the outside constantly. So that is the system which many freshwater protists, protozoa, will use in order to deal with the osmotic pressure, the continual osmotic pressure. But there are many other organisms which live in freshwater that we need to think about. And of course, if we want to think about animals who are entirely immersed in water, then of course the fish are a particularly interesting group. for all sorts of reasons. First of all, the fish evolved in freshwater systems, not in the sea, but in freshwater systems. And they carry an evolutionary history of that in their physiology. That's the first thing to tell you. The second thing is, if you picture how a fish functions, a fish lives entirely surrounded by water. They are covered with mucous membranes, and they also have extensive gills here, which are immersed in the water. There's a huge surface area in the gills, which allows them to respire, to get oxygen and get rid of carbon dioxide. But that also provides a huge surface area of cell membrane, which is now actually surrounded by the fresh water. The interior of freshwater fish is actually quite salty. It has a high solute concentration. So in actual fact, their water concentration inside is much less than the outside. And they have to deal with the problem that they are continually flooded with water. In fact, they're in grave danger of drowning, of their physiological systems being overwhelmed with water and diluted to such an extent that they can no longer function. So they have to have special tactics in order to deal with it. Despite the fact that ions are relatively rare in freshwater, freshwater fish have ways of actively grabbing ions and pumping them into their blood to maintain as high a blood concentration as is needed for their physiology to continue. So that's the first thing. The second thing is they extract as much ion as possible from their food in order to maintain the high ionic content of the blood. The second thing is their kidneys are extremely good at recovering ions. The grave danger for freshwater fish is that they will void so much water that they will wash out, they will dilute and wash out all of the ions in their body fluids. So the kidneys are extremely good at recovering ions and ensuring that they can maintain a high ion content. But all of this is very, very metabolically expensive. The fish have water flooding into them through their entire exterior, through all of those mucous membranes that cover them. They also have water flooding in through the gills the whole time. And they have to actively pump water out with the kidneys in a very dilute form as rapidly as possible. Their kidneys work overtime, simply voiding water and trying to recover as many ions as they possibly can. So that's the situation for freshwater fish. It's actually quite a difficult situation. What about saltwater fish? Well, in actual fact, saltwater fish have an ion concentration, which is pretty much the same as freshwater fish. And it's a holdover of their evolution in freshwater. Their ion concentration is not the same as seawater. They are not isotonic with seawater. In fact, their salt concentration is much lower than seawater. It's about half of seawater. What does this mean? Well, it means that exterior to them, the water concentration is much lower, and osmotic pressure is going to lead to them losing water. Saltwater fish actually dehydrate in saltwater. They dehydrate in seawater because they continually lose water from osmotic pressure. As a result, what saltwater fish have to do, seawater fish, they have to drink as much water as possible. So they continually drink, and they actively excrete the ions. They excrete ions from the gills. They have special pumping mechanisms to get rid of the ions, especially in seawater, sodium chloride, to get rid of sodium chloride and all the other ions that constitute seawater. And in addition, they excrete very, very concentrated urine. They recover as much water as they possibly can from the water, that seawater that they drink, and they excrete very, very concentrated, salty urine to get rid of it. So two contrast, despite the fact that they have very similar body fluids, two contrasting kinds of strategies that they need to adopt. How about plants? Well, here, I'll tell you, I have hinted at this already, but I'll tell you about this because you may, I think, do this in lab. This is a very common freshwater plant called Elodia. If you've got an aquarium, you almost certainly got Elodia. It's this kind of fluffy, looks fluffy kind of underwater plant. So it's immersed completely in fresh water. And under normal circumstances, Elodia allows its central vacuole to fill and to create a turgor pressure that presses up against the cell wall. And this is the normal situation here. If we now move this plant into a solution where the ion concentration is higher, in other words, the water concentration outside is lower, then what begins to happen is water begins to flood out of the cell, especially water begins to move out of the central vacuole, and the central volume of the central vacuole begins to decrease. It shrinks, and the cell begins to pull away from the cell wall. You can see it quite clearly, and you can see it quite clearly here as well, how it's pulled away entirely. And this we refer to as plasmolysis. Look here at this here, and you can see it. Here's the cell wall here, there. And here's the cell itself. It's forming a ball in the middle. You can see its distribution because you can see the chloroplast in the cytoplasm. But you can see where the cell membrane is around the outside there. And it's forming a ball, and that is because the central volume of the central vacuole has decreased so much. This is a reversible situation. As long as you don't leave this too long, you can put this back into the correct concentration of water, and the central vacuole will very rapidly increase back to its normal size until its turgor pressure has pressed up against the cell wall once again. Okay, so we've talked now extensively about the movements of water across the membrane under these different circumstances. What about all of the other stuff that needs to move back and forth across the membrane? Well, there are two particular circumstances that we need to think about. Remember that I told you earlier in this class that the ions behave independently of one another. So, for example, if I've got a high concentration of sodium on one side of the membrane, and I've got a low concentration of sodium on the other, and I need a higher concentration on the inside, then all I have to do is open a channel, a sodium channel, and allow the sodium to move. And it will move in the direction of its concentration gradient. It will move across the channel towards the inside and it will to equalize the concentration on one side and the other. And this is exactly what the cell often does, where there is a situation where the concentration is high away from where the ion is needed and low where it is needed. We can use that diffusion pressure to move the ion in the direction that we want it to go. And this, by opening the channel, this channel that opens is usually a protein, a specific protein, a protein specific for that particular ion. And because what is moving the ion is diffusion, we call this facilitated diffusion. Under normal circumstances, this diffusion would not occur very easily because there's a membrane in the way. but we open a channel specific for that ion, and we allow that diffusion to occur. So we refer to this as facilitated diffusion, and this is done by sets of specific transport proteins, which provide corridors across the membrane in order to allow ions to move down there, easily move down their concentration gradient. So these are systems of ion channels, especially, that we need to consider. These systems of facilitated diffusion, there are two ways in which they can actually operate. The first is the one that I've already referred to. It's by the existence of channel proteins. By the way, these channel proteins can be very rapidly, these channel proteins can be very rapidly created and embedded in the membrane if they're needed. So this is a dynamic system. The cell can respond to a need by creating these channels if it needs to do so. So here's a typical channel protein. It's very specific. It's specific only for one substance, for one solute. But it will allow that to move across quite freely. And it's simply like a tube through the cell membrane to allow it to move from one direction to the other. The important thing is that this is taking place in the direction of the concentration gradient. It's going in this direction only. There's no movement in that direction, because what is driving this is the diffusive pressure. The concentration of the substance is higher this side than it is that side, so it moves in this direction. Once this equilibrium, once the concentrations are equal on both sides, then net movement stops because it enters equilibrium. This is really important to realize. The system only works as long as there's a high pressure, high diffusive pressure on one side than there is on the other. The other kind of facilitated diffusion is undertaken by these sets of proteins, which are called carrier proteins. This one's a channel protein. This is carrier protein. And carrier proteins are somewhat different. They still operate, there's still a facilitated diffusion. It still operates in the direction of the concentration gradient. You've got a high concentration of solute here, low concentration here. But the carrier proteins actually bind the substance. They have a site which matches the structure of the ion that they're going, or substance that they're going to move across. And when that binding takes place, the shape of the protein changes. So they open on one side, they bind, they change shape, and they spit out the substance on the other side. But all of this activity is driven by the diffusive pressure. It's driven by the need to equalize the concentration on one side and the other. There is no additional energy required. As long as there is a diffusive pressure, the protein will carry on moving the ion from one side to the other. So that's facilitated diffusion. Very commonly, however, the cell is going to need to accumulate substances in high concentration inside the cytoplasm, or maybe inside an organelle. The cell is going to need to accumulate substance and maintain the high concentration against a concentration gradient. Then you're going to need to grab stuff which is relatively rare and move it to an area where it's relatively abundant. This is quite common. The cell is going to have to do this all the time. And if the cell needs to do that, it is going to need to put in energy. Whenever we need to move a solute against its concentration gradient, we have to put in biologically useful energy. And that energy is derived from ATP. And so these systems are driven by biologically useful energy. I say always by ATP. Actually, there's one or two which use other sources of energy, but it's always a phosphate. So ATP is adenosine triphosphate. There are one or two other substances which also carry the phosphate group, but by far the majority are driven by ATP. These are systems of active transport. They require the input of energy. And in doing so, they hydrolyze some substance which is high in phosphate. They very often are themselves phosphorylated. That's how they acquire the energy. They become phosphorylated and they acquire the energy, which is potential energy in that phosphate bond. So they are, in fact, carrier proteins in that they change shape. So it's similar to the facilitated diffusive carrier proteins. They actively change shape, but the change in shape is driven by the binding of the substance and by their phosphorylation. So here's a typical situation, and this is a common one, by the way. This is very important in many cells, especially important in nerve cells. And this is a situation that arises because often the cell needs to maintain a high concentration of sodium salts outside and a low concentration of sodium salts inside, and the opposite for potassium. It needs to maintain high potassium inside and low outside. So this system, this protein binds two things. It binds sodium and it also binds potassium. And it's going to move them actually in opposite directions. In order to do that, it's going to require an input of biologically useful energy. And that comes in the form of ATP. So here is the carrier protein here, and it's embedded in the membrane as normal, and it's a transmembrane protein. So here it is facing into the cytoplasm. And it has these three sites here, which bind sodium ions. And when it has bound 3 sodium ions, it becomes susceptible to phosphorylation. This phosphorylation will not occur until these 3 sites are all bound. It's inherent in the nature of this transport protein, which is, by the way, at the same time, it's an enzyme. But the protein itself is phosphorylated by ATP. It hydrolyzes ATP. It takes off one of the phosphates, and that phosphate becomes bound to the protein. And as soon as that happens, the protein changes shape, and it opens up to the outside. At the same time, that shape change means it can no longer hold on to the sodium ions. The shape change is such that the sodium ions can no longer bind to the protein, and they get spat out. But at the same time, the shape change opens up 2 sites where potassium can bind, and potassium from the outside now binds to the protein. When that happens, the protein becomes unable to maintain its phosphorylation, and the phosphate group is lost. And as soon as that phosphate group is lost, the protein returns to its original shape. And when it returns to its original shape, the shape of the protein changes such that it can no longer hold on to the potassium. It spits the potassium out. And now it's back to where it was before. Its sodium sites now open. Its potassium sites are closed. Fills the sodium sites. It changes shape. It becomes phosphorylated and changes shape. Spits the sodium ions out so that they begin to get accumulated on the outside relative to the inside. And it acquires potassiums. It dephosphorylates and reverts to its original shape and spits the potassiums out in this direction. It carries on doing this until it reaches a situation where concentration of potassium is high inside, the concentration of sodium is low. And the concentration correspondingly, the concentration of sodium is high outside, and the concentration of potassium is low. Now, this is for all sorts of reasons. This is especially important, as I told you. It occurs in many different cells, but it is especially important in nerve cells. Because remember what I told you in our class when we were talking about how charge is so important. The difference in these two ions, actually, the difference in concentration of those two ions leads to what is effectively a charge difference across the membrane. But second of all, it leads to this double difference in concentrations across the membrane. You've got a high diffusive pressure here for sodium, a high diffusive pressure here for potassium. And the cell is going to use those. It's going to allow those, in fact, to equalize. And when they equalize, it's going to extract energy and use it to do work. That's why this sodium-potassium pump is so important. As soon as the work is done and they've equalized again, the pump gets working again, and it reestablishes these gradients in order so that the cell is ready to perform its next action. Okay, so we'll finish off there for the time being. Please remember, go through this diagram, which provides you with a summary of the whole system, of all these systems. We have a membrane, which is semi-permeable. It allows very few things to diffuse across it very easily. Instead, the cell embeds within the membrane proteins which allow the cell to become semi-permeable. That is, to control what ions and what substances move across the membrane. It, in fact, is exquisite control. The cell is very good at controlling what goes across. That transport may be by diffusive means. It may be facilitated by the proteins, but not require energy as long as it's moving substances from high concentration to low concentration. But very often the cell needs to move substances against their concentration gradient. And when that is required, the cell has to put in energy, and those are the active transport systems. Any protein which changes shape in order to do this is called a carrier protein. Any protein which binds a substance and changes shape to do so is called a carrier protein, whether it's facilitated or active. But in the active transport systems, this protein will usually do so by becoming activated and phosphorylated from ATP. All right, so we'll leave it there, and then I'll see you next week.